Hello everyone, very warm welcome everyone, very warm welcome to ITLS Academy. The topic of today's lecture is repairing of heart by stem cells. Let's start with introduction. First point, for those suffering from common but deadly heart diseases, stem cell biology represents a new medical frontier. Researchers are working towards using stem cells to replace damaged heart cells and literally restore cardiac functions. Next point. One of the major contributors to the development of ineffective pumping conditions. Ineffective pumping conditions means when heart is not pumping properly, when blood supply is not proper. This condition is known as ineffective pumping and it results into a heart attack. Heart attack in which there is no pump, there is ineffective pumping. We cannot say that there is no pumping. There is pumping, there is blood is coming, but that coming blood is not sufficient. That is the ineffective pumping. This heart attack, known medically as a myocardial infarction, sometimes it is also known as chest pain. Chest pain, medically known as myocardial infarction, while no pumping condition, this is the heart attack. It is easy to recognize that impairments of the heart and circulatory system represents a major cause of death and disability in the United States and also in our nation. Next point. Despite advances in surgical procedures, mechanical assistance devices, drug therapy, and organ transplantation, more than half of patients with congestive heart failure die within 5 years of initial diagnosis. Researchers are now exploring ways to save additional lives by using replacement cells for dead or impaired cells so that the weakened heart muscle can regain its pumping power. Here in this diagram, you can see this is rodent model of myocardial infarction, or we can say this is of a model of heart attack. In this diagram, you can see this is heart of mouse. This is dissection of mouse heart. The dissect of mouse heart is taken here. You can see this is coronary artery ligated, inducing and infarct. This is infarct. This is how myocardial infarction occurs in the rodents or in the mice or in the case of mouse. How might stem cells play a part in repairing the heart? How stem cells can repair the heart? What role they have in case of repairing? Let's see what are the answers of all these questions. First point, one important type of cell that can be developed is the cardiomyocytes. This is cardiomyocyte. As we know that cardio word is taken for the heart, it is used for the heart, and myo word is used for the muscles. So what are the cardiomyocytes? These are muscles of the heart. The heart muscle cells. Sight word, as we know that it is used for the cell. So these are heart muscle cells that contracts to eject the blood out of the heart's main pumping chamber. For the pumping, these heart muscles are responsible. So where is the role of these stem cells? Where these stem cells playing a role in the repairing? This is the process of heart. So to answer this question, researchers are building their knowledge base about how stem cells are directed to become specialized cells. If we talk about the heart muscle cells that are responsible for the pumping and heart attack, the main reason of heart attack is the loss of ability of pumping. So this may be a huge step if we will be able to replace these heart muscle cells so that we can restore the pumping ability of our heart. As we know that the heart has a large demand for blood flow, and these specialized cells, that is heart muscle cells, are important for developing a new network of arteries to bring nutrients and oxygen to the cardiomyocytes. After a heart has been damaged, coming to the next point, the potential capability of both embryonic and adult stem cells to develop into these cell types in the damaged heart is now being explored as a part of a strategy to restore heart functions to people who have had heart attacks or have congestive heart failure. Injured heart cells can shift from a quiescent state into active cell division. What is quiescent stage? Do anyone know what is this quiescent stage? Okay, I will tell you what is this quiescent stage. As you all have studied the cell cycle. Cell cycle have three phases, three main phases: G1, this is G1, this is S, and this is G2. They have a very small part of M phase. The cells that are going through this cell cycle divide continuously, and they are divided continuously. But some cells leave this cell cycle and become specialized, that in the phase of a phase you may know. This is G1 or G, this is G0 or G0 phase. This G0 phase is known as quiescent phase. So we can say this phase is a specialized kind of phase, because all these cells that are present in G1, G2, S or M phase, they are a dividing cell, but the cells in the G0 phase are a non-dividing cell. The cells present in the G0 are non-dividing cells. They are not dividing, but it doesn't mean that they will never divide. These cells divide whenever there is an requirement. They de-differentiate again, and these can help in forming of new cells. So here, if we will able to generate these cell for the de-differentiation, so we can restore heart functions by the help of these cells that are in the quiescent stage. Researchers now know that under highly specific growth conditions in laboratory culture dishes, stem cells can be coaxed into developing as new cardiomyocytes, that is heart muscle cells, and vascular endothelial cells. Vascular endothelial cells are a kind of cells that are involved in the formation of the wall of blood vessel. If this is a blood vessel, so what is the function of this blood vessel? Do anyone know? Okay, now I will tell you. These blood vessels are the main capillaries that are responsible for the transport of blood from one place to another place. The wall of blood vessels, this is the wall of blood vessels. Let me draw with another color. The wall of blood vessel is made up of some kind of cells. This is the wall of blood vessels. It may be thick in the case of it may be thick, and it can also be non-thick. So this wall is made up of the cells that are known as vascular endothelial cells. So we can say the endothelial cells are the cells that are responsible for the formation of wall of these blood vessels, not blood vessels, wall of these blood vessels. Scientists are interested in exploiting this ability to provide replacement tissues for the damaged heart. to the next point, a question arises here. What is the evidence that such an approach to restoring cardiac functions might work? This question is obvious. How we can say that this will work? So in the research laboratory, investigators often use a mouse or rat for the study and investigations. And these rat models are a very suitable organisms to study the function of a heart attack, to study new therapies. During the past years, researchers using such models have made several key discoveries that kindled interest in the application of adult stem cells to heart muscle repair in animal models of heart disease. There was an experiment that was performed on the mice. So I think I must tell you that experiment, although I have not mentioned that experiment in this slide. So I am telling, a heart attack was induced in mice by tying off a major blood vessel. Okay, the left main coronary artery, that was that blood vessel that was tied. This was coronary artery. Through the identification of unique cellular surface markers, I think you all know what are surface markers, because I have already discussed in our previous lectures. So by the use of surface markers, the investigators isolated a selected group of adult primitive bone marrow cells with a high capacity to develop into cells of multiple types. What are those kind of cells that can develop into multiple types? I hope you all know. These are pluripotent kind of cells. When these cells injected into the damaged wall of the ventricles, these cells led to the formation of new cardiomyocytes, that is, formation of the new heart muscle cells. Apart from this, there was formation of vascular endothelium and smooth muscle cells, thus generating the new myocardium, including coronary arteries, arterioles, and capillaries. So this is our experiment. I have put a slide. I didn't know. Okay, let us see what was this experiment. This is mouse heart. I hope you can see. Mouse adult stem cells are injected into the muscle of the damaged left ventricular wall of mouse heart. The mouse adult stem cells. These are adult stem cells. These are injected into the left ventricle. What happens here? These adult stem cells started dividing. You can see here, these started dividing. Stem cells help regenerate damaged heart muscles. As these these adult stem cells started to proliferate, started to uh, differentiate into various types, and they started the formation of new heart muscles. These are new heart muscles. You can see these are damaged heart muscle cells, but these cells are new cells that are restored by these adult stem cells. On the other hand, human adult bone marrow stem cells, these were mouse adult stem cells, but in the second case there were human adult bone marrow stem cells, and these are injected into the tail vasculature of a rat. These are injected into the tail. These are adult bone marrow cells of human. The stem cell induce new blood vessel formation. Here, new blood vessel formation started in the damaged heart muscle and proliferation of existing vasculature. Here, proliferation of the existing vasculature has started. You can see here these are adult stem cells, and these started proliferation. These are damaged heart muscle cells, and these are new blood vessels that started forming into the mice. So experiment was successful. Heart muscle repair with adult stem cell. This is the, if I give a headline to this particular image, so it will be heart muscle repair with adult stem cells. The experiment was very successful, and it was clear that we can use adult stem cells for the regeneration of heart muscle cells as well as for the formation of new blood vessels. Because of their ability to differentiate into any cell type in the adult body, embryonic stem cells are another possible source population for cardiac repair cells. As we have studied embryonic stem cells, these cells are pluripotent in nature. And we know what are pluripotent cells. Pluripotent cells have the ability to differentiate into various kind of cells. So these were another possible source from which we can restore heart muscles. So there were an experiment with the human embryonic stem cells. In the first step in this application, who demonstrate that human embryonic stem cells can reproducibly differentiate in culture into embroid bodies made up of cell types from the body's three embryonic germ layers. Three embryonic germ layers are ectoderm, endoderm, and mesoderm. These embryonic stem cells can form all these three layers. In a continuation of this early work, a scientist was Kehat and his colleagues displayed structural and functional properties of early stage cardiomyocytes in the cells that develop from the embryoid bodies. The cells that have spontaneously contracting activity are positively identified by using markers with antibodies to myosin heavy chain, alpha actinin, dasmin, anti-natriuretic protein, and cardiac troponin, all proteins found in heart tissues. These investigators have done genetic analysis of these cells and found that the transcription factor genes expressed are consistent with early stage cardiomyocytes. These breakthrough discoveries in rodent models present new opportunities for using stem cells to repair damaged heart muscle. The result of the studies discussed above are growing evidence that adult stem cells may develop into more cell types than first thought. Although there is much excitement because researchers now know that adult and embryonic stem cells can repair damaged heart tissue, many questions remain to be answered before clinical applications can be made. For example, how long will the replacement cells continue to function? Some other questions like do the rodent research models accurately reflect human heart conditions and transplantation response? Do these new replacement cardiomyocytes derived from stem cells have the electrical signal conducting capabilities of native cardiac muscle cells? All these questions are arises. So we can say for what is the answer to this question? Stem cell may serve as the foundation upon which a future form of cellular therapy is constructed. In the current animal models, the time between the injury to the heart and the application of stem cells affect the degree to which regeneration takes place, and this has real implications for the patient who is rushed unprepared to the emergency room in the wake of a heart attack. Here you can see how stem cell therapy is applied. This is a mouse and this is mouse heart. This heart is not healthy. This is a damaged heart. Here is cardiac stem cell injection. After injection, myocardial repair and regeneration was seen. This was limited cardiac stem cell survival, partial cardiac stem cell derived remuscularization, and incomplete maturation of remuscularized myocardial tissues. On the other hand, these are cardiac stem cell loaded 3D scaffold injections. Again, there was myocardial repair and regeneration was seen, and this is the dissection of this particular part. You can see in this extensive cardiac stem cell derived remuscularization of the infracted tissue after improved survival, differentiation, and maturation of injected cardiac stem cells. So thank you very much guys for watching this session. Follow ITLS Academy on various social media platforms. Thank you very much everyone.